Good morning. My name's James Danger, and this video is on a really fantastic paper I've just read called Finding the Right Sperm, Let the Female Tract Decide. For me, it was quite revolutionary. After 50 years of IVF, I thought this was just an eye-opener for a paper. Okay, so the paper was published in Animal Reproduction. This is really an animal experimental reproductive andrology paper. It was presented at the Brazilian Embryo Transfer Society, which is really to do with domestic animal transfers, but it's a fantastic one that applies equally to laboratory, domestic animals, and to us mere mortals. So this is really a tale of the assault by a large army of millions of sperm on a tortuous journey, narrow passages, imprisoned in the tunnels, released to the sweet adjustments of perfumes, and a final acceptance of the prize by the chosen one. No, it's not the Odysseus. It's called Sperm Selection in Vivo or in real life. And this is the story. And it's a fantastic paper. It really is worthwhile reading. In vivo mechanism for sperm selection female tract, uterine clearance mechanisms, the uterotubal junction, the formation of an oviductal sperm reservoir, sperm release from the reservoir, sperm navigation in the oviduct, mimicking the female tract to improve in vitro embryo outcomes, microfluidic platforms for active biomimetic sperm selection, oviduct epithelial and ligand-mediated sperm selection, and conclusions. And of these, I'm going to go through all these chapters very quickly in the paper. I'll try and make it not too long, and then have my own views, of course, at the end. But I have to reckon, like all papers, there are thousands of articles published in IVF each year. This is just the number of papers in IVF year by year, and this is one for PGT-A. You can see how quickly they rise. It's impossible to keep track of them all. And so with the papers that I select from my YouTube channel, the ones that I think are important, both for IVF staff and their clients. And like always, I've a notice board where if you're interested in promoting your product or clinic, this is a space you may like to quickly put a notation of what you do. And so you can contact me at Third Aid if you're interested. Okay, enough of that rubbish. Let's get on to the subject at hand, the story. So each of these is a subject heading this paper. It's open access, so you can read it. And I actually think there's a lot of really interesting stuff. And if you have an open mind about sperm selection, then this is a good paper. There's been a lot of work done just recently showing there's a lot of interaction between the sperm and the oocyte, and that one of the problems with eggs from older ladies is they don't have the capacity to correct potential damages in sperm. And a lot of what happens in the first 24 hours of the zygote's life has direct implications the formation of the inner cell mass and implantation. So selecting the right sperm is important. In the past 30, 40 years, I think we've been fairly ignorant and even vain in thinking that what we do in vitro makes any difference. This paper has changed my views quite a lot. And a lot of things which we look at, like polyspermy and sperm DNA, don't occur in nature. It may occur in the sperm sample, but it doesn't occur in the fallopian tubes. And so if I've been influenced significantly by this paper, I hope you are so as well. If you're not, well, fair enough. Let's just go through it. Okay. So the sperm used in IVF is in many ways an artifact of the in vitro technology. Almost nothing about in vivo sperm selection is replicated in vitro. Almost nothing we do with sperm preparation in vitro is remotely like what happens in vivo, mainly because we just don't bloody know what happens in vivo. It's been a black box we've turned our back on, and there's now sufficient amount of data to suggest that, in fact, what happens there is even more exciting than the movie. Okay. The sperm almost never ever sees an abnormal sperm. And so why is it surprising that the capacity to fix fragmented sperm in zygotes is very limited? Because normally it just doesn't happen. And polyspermy, according to this article, almost never happens. Right? So we see polyspermy in IVF and we think it's to do with the eggs and sperm, but in reality, that doesn't happen in nature. The sperm selection process is so tight that in reality, polyspermy is an artifact, is an in vitro artifact. Another thing too is sperm in the oviduct tract are held in an arrested state, whereas in vitro, the sperm are washed and they're free to run around as much as they like kids in a playground, right? But that's not what happens in vivo. That's very contained. They're held in an arrested state to mature. They're only released at the time of ovulation when there's an egg there. They're released with chemical messages, and the capacitation occurs only on release, whereas in vitro, as soon as they're washed free of seminal plasma, that's what I did my PhD on in 1970, sperm capacitation in the mouse. As soon as you wash free of the seminal plasma, they start to capacitate spontaneously. They're going all over the place. We've been preoccupied with, no, actually no one ever really cares about capacitation, but they should. But in vivo, it's a really part of a ritual that sperm have to go through, that the egg requires the sperm to go through in order to be fertilized by a good sperm. The selection by a number of physical and chemical signals. And if this paper doesn't change your mind, then maybe I'm on a different planet to you. Okay. So the mechanism of sperm migration is generally difficult to study in situ because the female reproductive is inaccessible without invasive procedures. That is, you've got to kill the animal to look at it, right? Yeah. So almost all in vivo studies on migration of the oviduct provide static post-mortem or post-surgical images and do not accurately reflect the dynamic process involved. It's almost as if we just turned a blind eye. We said it's too hard. We're just going to ignore it. We don't need to worry about it. Let's get on with life. The blastocyst and PGT and euploid embryos are more important, but this at the start is just as critical. Furthermore, the majority of sperm in the oviduct are bound to the luminal epithelium or very deep within a very narrow mucosal folds, which underestimates the recovery rate. Thus, the in vivo sperm counting data should be interpreted with extreme caution and is almost certainly wrong. Further analysis of sperm retrieved in the uterotubal junction, the UTJ, and the oviducts of pig and mice indicate that more than 95% were normal morphology with an intact acrosome. Now, in all fairness, most domestic animals and most mice are inbred strains with high fertility, and so we actually don't know what happens in humans. And it may well be that all the rubbish we go on about with sperm morphology is actually kind of irrelevant, because what happens in the uterotubal junction might only be the normal sperm, of which there's more than enough to do the job. It's kind of sad to think my whole PhD was based on sperm morphology and stuff, and it's kind of rendered irrelevant. The uterine, now the first thing is getting rid of all the rubbish. We do that by putting it on a centrifuge gradient and getting rid of the overlays, or doing a swim up and getting rid of the pellet. But what happens in utero is there are uterine contractions that increase during estrus and may also be further stimulated by the presence of sperm. A substantial portion of sperm is eliminated by retrograde uterine contractions with 1 to 2 hours post-insemination, a process called semen backflow. And that doesn't apply to humans, of course, but most of the sperm don't get out of the vagina, right? Retrograde flows contain normal and motile sperm and seem to be enriched in abnormal sperm containing cytoplasmic droplets and tail defects, as compared to the ones found in the tip of the uterine horn. That is, the sperm that are washed out with retrograde flows are all the abnormal sperm, poor motility, anything to do that is not a really healthy sperm. So about all the rubbish we apply, this suggests that there is an initial selective process, not unlike, for instance, the first process where you do density gradient centrifugation within the female reproductive tract. All the rubbish we apply to sperm morphology, while it's an important diagnostic tool for male fertility, male health, is not an issue in vivo. These sperm are just removed, or so we're led to believe by the story. The remaining spermatozoa are perceived as foreign invaders in the uterus and trigger a maternal innate immune response involving macrophages and polymorphonucleoside neutrophils, or PMNs. In cattle, PMNs increase within 2 to 6 hours after AI in the uterine lumen and eliminate sperm through direct phagocytosis and the formation of neutrophil extracellular traps. Okay, just move on from that. The incubation of sperm within the PMNs trigger these ETS formation within 3 hours, resulting in a net reduction in progressive motility compared to controls in humans. So this is something which has been observed with humans, and maybe one day I might do a video on it. In vitro studies in cattle and humans indicate that these polymorphonuclear neutrophils target sperm with a premature capacitation rather than non-capacitated ones, suggesting this is a second selective process of uterine clearance. So we don't actually think of all these sperm which are being prematurely capacitating, but the neutrophils, the argument goes here, is that they are all being selectively removed because they have actually already undergone the acrosome reaction or some form of change in their morphology and their external morphology. But the thought also occurs to me, this may also be an important immunological signal the female notes that intercourse has occurred. Spelling mistake. Something may be missing in IVF. In other words, just 1 or 2 days before, or maybe of the night, I have no idea. That intercourse actually is a signal that the female can use to say intercourse has taken place, and that may trigger some form of uterine maturation that we're not really aware of. And maybe an IVF where we're telling people to abstain for 2 or 3 or 4 days might actually be the worst thing we can do. Maybe we should tell everyone to go home and have intercourse the day after pickup. I'm sure that won't go down well. Okay. The uterotubal junction represents the first major anatomical barrier encountered by sperm inseminated in the uterus. This region is characterized by a significant constriction in diameter combined with a maze-like arrangement of mucosifieds that extend into the lumen and mucin-rich viscous secretions, which together form a stringent physical barrier to sperm aggression. You can see here, if your brain is thinking wisely, that this is where you can start to create the microfluidic devices. Make it hard for sperm to get through the device. Beyond the physical barrier, sperm must present a molecular passport to be able to cross the uterotubal junction, at least in mice. Now, this is something which I've never come across before. To date, 27 genes have been identified in mice as critical for sperm to reach the ovum. Now, I've never in my 50 years of IVF have thought that there is a genetic makeup that allows sperm to progress. It's not part of our thinking. It's changing. So the authors go on and say, male mice lacking any of these genes have severe infertility despite exhibiting normal sperm morphology and motility, and underscoring the importance of specific molecular interaction with the female tract. Along with these genes, 22 are involved in the expression, maturation, and membrane localization of disintegrin and metalloproteinase 3 or ADAM 3. Now, I have to say at this point, if many of the viewers are like me, I get kind of turned off when we start talking about molecular biology and sperm and ADAM 3s and ADAM 5s, etc. But this paper has kind of changed my mind a bit and making me realize, in fact, these are part of the keys to the door that sperm need to demonstrate. But they also give us a clue that how we may be able to, in the future, molecularly interact with sperm to select in vitro the ones that are being selected in vivo. I'm amazed to think that there is a molecular key that sperm have to have in order to progress up the tube. That's really quite a hard concept for me to get my head around. The protein ADAM 3 is also needed for sperm binding to the zona pellucida, indicating a shared mechanisms between sperm migration to the oviduct and the ability to fertilize oocytes. At the molecular level, the sperm surface protein ADAM 5 among proteins needed to cross the uterotubal junction are shown to interact with laminin 1 and fibronectin, two proteins primarily present in the extracellular matrix, but also report on the surface of the oviduct lumen. So I need to pass over this because there's a lot more stuff to get through, but I think we need to come back to this and think a bit more about a molecular passport. This molecular passport may prevent unfit spermatozoa, as well as those undergoing premature capacitation from reaching the oviduct. In mice, after mating, sperm in the isthmus exhibit a high proportion of intact chromosomes and lower tyrosine phosphorylation levels, a marker of capacitation, compared to those in the uterus. And now, get this is what caught my eye. The selection by the UTI may extend to chromatin quality. Spermatozoa recovered from the oviduct exhibit low DNA fragmentation to those in the uterus. Wow. Together, these findings highlight the role of the uterotubal junction as a multi-level gatekeeper against sperm with structural, functional, and DNA abnormalities. Now, remember, this is in mice, and it doesn't work the same way as in humans, but it may well be that, in fact, the same mechanisms apply, but in a different way. But all the work we do in sperm DNA fragmentation and trying to work out this and that, whatever, because I've spent a lot of time running EQA programs for sperm DNA fragmentation. When I read that, it changed my mind that, in fact, the binding of sperm to the uterotubal junction is such a high selective process, and already we're getting rid of sperm DNA fragmented sperm. This is why I said in vivo eggs may never see a sperm with high DNA fragmentation. So there are two figures. I'll just go and read the, you can look at the picture while I talk. This is the mechanism of sperm selection through the uterotubal junction. It acts as a major anatomical molecular barrier regulating sperm entry into the oviduct. Its narrow lumen and complex mucosal folds restrict sperm passage, allowing only a small subpopulation to progress. In mice, sperm transit through the UTJ depends on specific behaviors, including the formation of sperm clusters, which involves interaction between sperm surface proteins and the epithelial ligands. Of course, look at all these. This starts to, I don't get switched off when I see this. But in fact, I think I'm going to have to learn my switch on my radar to pay more attention to it. And the second figure, the mechanism of sperm selection through the formation of a reservoir and release towards the oocyte in the cow oviduct. Okay. After crossing the uterotubal junction, a subpopulation of spermatozoa binds to the ciliated epithelium of the isthmus, forming a functional sperm reservoir. So not only do they have to get through the key, through the uterotubal junction, but once they're through, then they start to collect by binding to the oviductal epithelium, forming a reservoir. Binding is mediated by interactions between sperm proteins and the oviduct epithelial components, including annexins, fibronectin, and glycan motifs. Sperm release is triggered. This is what I think is really important. This, I think, is the crux of what we need to be focusing on IVF going forward. Sperm release is triggered by the periovulatory signals that promote detachment through hyperactivity, motility, and progression towards the fertilization site. So in other words, these sperm get through the uterotubal junction with their passport intact. They then bind, and I don't know how long they can stay there for. They suggested 2 or 3 days. And they have to wait for the ovulation to occur. And as the follicular fluid passes down through the tube, the media rich in progesterone, but maybe many other things that we haven't yet identified, causes those sperm to detach, can commit capacitation and hyperactive motility. And I just think it's amazing. Okay. After crossing the uterotubal junction, sperm reach the isthmus, the first part of the oviduct, where a subpopulation binds stably by their heads to the luminal cilia, forming a functional sperm reservoir. Several studies reported that only sperm with active flagella beating and presenting an intact acrosome with no morphological abnormalities are able to bind to the oviductal epithelium. Now, I don't know how accurate they are, etc., but this is the intention that the sperm selection process is for the really best of the best sperm. Beyond expecting sperm lifespans, the sperm reservoir is thought to synchronize gamete encounters and reduce the risk of polyspermy by facilitating the gradual release of sperm towards the ampulla. Okay, the time release of spermatozoa, we're getting closer to the fertilization here. The time release of spermatozoa from the reservoir towards the ampulla, where the cumulus oocyte complex advance following ovulation, is essential for fertilization. In cattle, there is evidence that sperm release from the oviductal epithelial cells is mediated by progesterone through membrane receptors and cat sperm-dependent calcium influx. The oviduct is a manure-inducing sperm capacitation, a series of metabolic and physical changes leading to the acrosome reaction. Now, all my life, I have always considered the capacitation to be a natural release from seminal plasma. But this story is quite different. This is telling how the sperm can get out of the seminal plasma, penetrate through the uterotubal junction, be bound by the epithelia, and remain in a non-capacitated state, almost dormancy, until required. And that's something that even when I did my first mouse IVF back in 1977, right, that was never part of my thinking. In vivo imaging of mouse sperm progressing through the oviduct showed an increase in the proportion of sperm with reacted acrosomes from 2% of the lower isthmus to 95% in the ampulla. However, it is likely that release from the reservoir is linked to the initiation of capacitation. Now, most people in IVF have done away with the concept of capacitation. There's been almost no discussion or papers written on it for the last 10, 15 years. It seems to be an issue about sperm that no one really cares anymore. It's not an issue with ICSI, who cares? But here we are in life, after every intercourse event. The sperm are bound to the epithelium, and the capacitation has only started when they're released from the reservoir. The comparison between bound released and controlled unbound bull sperm evidenced amongst the released population a higher progressive motility, elevated intracellular calcium, and higher membrane fluidity, all signs of ongoing capacitation. The sequence of events at the moment of release is, however, not entirely clear. Progressive capacitation of some sperm may trigger their release, or female signals trigger first release, which in turn causes capacitation. The sequence of events at the moment of release is, however, not entirely clear. It seems that whiplash motility may be involved in the release, and so, at least to my mind at least, the whiplash motility may seem in vitro to be an artifact. Moving on, the proposed model of sperm guidance in the cow oviduct. Spermatozoa navigate through the oviduct by integrating multiple physical and chemical cues. Now, this is getting to the really important part, and this is where we'll start to look at new microfluidic devices. Rheotaxis, orientation against the fluid flow. Thigmotaxis, a wall-following behavior. Thermotaxis, migration along temperature gradients. And chemotaxis, response to chemoattractants. Now, I've been aware of these all my life, but suddenly I think we need to read back and focus on this. Several facts prompted the search for mechanisms to guide sperm towards the fertilization site. First, the number of sperm reaching the isthmus reservoir is, as seen above, very small. Second, the distance between the isthmus and the ampulla is relatively long, about 15 to 20 centimeters in cattle. The overall length of bull sperm being on average 70 microns, this path is 2,000 to 3,000 times longer than the length of the sperm. Third, the oviduct lumen is a labyrinth filled with highly complex mucosal folds, at the bottom of which one egg cell in humans and cows may be hiding. Lastly, matured oocytes, and this I think is important, have a very limited period of fertility, estimated around 6 hours in cows, which means that sperm guiding should be efficient and relatively rapid, especially if insemination is performed close to ovulation time. And one of the things I should have had a slide here is that in vivo, fertilization doesn't occur at 38.5 hours after HCG. Ovulation occurs somewhere from 36, 37 hours all the way to 42, 43, 44 hours, because we do our egg collections before the first follicle is ovulated, and then we let the eggs mature in vitro. But in vivo, ovulation can occur over a 6 or 8 hour period, but it's only once the follicle is burst and the follicular fluid runs down the oviduct containing progesterone and many other factors is the sperm triggered. So in fact, the sperm sit there waiting until the oocyte has been released. And that is a variable thing. It's highly tuned and probably synchronized. And I think that's probably one of the really important things. There is evidence that sperm are able to orientate and swim in response to some physical and chemical external stimuli. These particular behaviors include rheotaxis, thigmotaxis, thermotaxis, and chemotaxis. Thigmotaxis is defined as a behavioral trait characterized by the avoidance of open areas, a bit like me in a way, preferring to remain near walls like rodents, in open field tests. That is, they swim against the edge of a wall rather than just randomly. So when you see sperm swimming on the bottom of a dish in IVF, that is an abnormal. People would argue this is abnormal. This doesn't normally happen. Sperm wouldn't swim randomly. They would be following the line of the cell wall bit by bit. And called thigmotaxis. Capacitated sperm is also guided by thermotaxis and chemotaxis. And sperm, get this, sperm are extremely sensitive to temperature variations. Human sperm can sense and respond to temperature differences as low as 0.01 degrees per millimeter. And the thermotactic responsiveness has been observed over a wide range of temperatures from 29 to 41 degrees. So in our IVF dish, the temperature is all the same. There is no thermogradient, and we have no idea how important that is to a sperm. But maybe, but that's part of our microfluidic device. And here's a fantastic paper published in Communication Biology in Nature, Sperm Navigation in Humans, a concerted effort of multiple means by Michael Eisenblatt from Israel. And I think that that's an article you should read. Thermotaxis can be considered as a long-ranging guide mechanism that guides sperm from the reservoir to the water ampulla. Sperm thermotaxis relies on a family of membrane G protein couplement that can sense light or temperature depending on cell types, and there's all these different things which I don't need to worry about. Opsin 2 and opsin 4 are shown to be crucial for sperm to perform thermotaxis, each mediating different cell pathways. And if you read this article, which is open access, you'll realize just how complicated it is. You'll never look at sperm in the same way again. Conventional mechanisms of sperm sorting before in vitro embryo production include density gradient, centrifugation, swim up. You're probably getting sick of my voice by now. In humans, and other sperm selection methods have been tested for IVF ICSI, including hyaluronidase binding assay and annexin 5 magnetic activation cell sorting, with inconsistent results. But I think we're looking at the wrong way. We're picking out the very best embryo to put back and saying there makes no difference here. And I think we should just forget all the IVF and think about the sperm. The simple and cost-effective swim-up technique remains one of the most widely used methods of sperm production. In fact, I used that back in 1977 for beginning my PhD and 1982 for my first IVF case, which I have to tell you started a pregnancy, and relies on sperm motility. In practice, and by the way, I might say here, when I first started IVF in 1982, I used to do swim ups, but I used to do it really quickly. I used to let the sperm only about 5-10 minutes at the swim up, and I would take the sperm off at the very top. Almost by the time I had a cup of coffee, then I'd take them off. And my first 19 pregnancies in IVF were all males. But as I went on and became busy, the swim up time became longer and longer, which I would then dilute. And I had more and more females. So anyone listening to this video, if there's one of a research project, you may like to ask the question, if you can do sex selection on individual sperm, look at the gender selection as they swim up over time from T0 onwards. And I would predict that the first 5 minutes of sperm, in the first 5 minutes sperm selection, more of those will be Y bearing than X bearing. Anyway, if you do that, send me an email if you do the experiment. But my first 19 and the first 20 babies were all males. Then slowly more and more females were born. So then about 30 minutes, most motile sperm recover. In fact, as I was saying, I used to do it in 3, 4, 5 minutes. The first sperm swim up the tube I grabbed and did IVF on. On the other hand, density gradient separation separates sperm based on morphology. The mechanism is essentially a physical filtration process based on cell density. Morphologically, normal sperm have a slightly higher density than abnormal sperm, and they pass through the gradient form a pellet, while lower quality sperm and dirty brood remain in the upper interfaces. But as you can see, this is from Eisenbach's paper, sperm sorting microfluidic chips based on sperm natural behavior in the female tract. These devices enable sperm sorting by exploiting different responses observed in female tracts. I can tell you by 2030, this will be the way that we will be doing sperm selection. If we're not doing that sperm selection by 2030, I think we're doing a disservice for infertile couples. So these are different ways that you can do sperm selection based on motility, chemotaxis, thermotaxis, or rheotaxis. Microfluidics have emerged as a promising strategy to improve conventional sperm selection methods. Microfluidic devices are also called microfluidic chips, are also defined as miniaturized systems that allow fluid control at the micrometric scale, typically in channels with dimension ranging from 10 to hundreds of micrometers. Microfluidic devices are very versatile, allowing for sperm selection based on a wide range of criteria. Another way to improve IVF is to rely on the capacity of some sperm to bind to the oviduct sperm reservoir. As stated earlier, only motile sperm with an intact acrosome and the absence of morphological abnormalities are capable of binding to an oocyte or sperm complex. In vitro models mimicking this reaction, including explants, epithelial monolayers, spheroids, and isolated oviduct-like ligands, consistently demonstrate that sperm binding competence correlates with membrane integrity, mitochondrial functionality, and DNA integrity. This suggests that adhesion is not merely a retention mechanism for sperm randomly captured by the cilia, but a biomarker of sperm quality. Additional studies in cattle reported higher cleavage rates following IVFs using sperm sorted by pre-binding to oviductal cells compared to control sperm lacking interaction. I should do even a video on that. Notably, the release of bound sperm was either induced by heparin, a mix of heparin and progesterone, or a mixture of heparin and COCs indicating that the peri-ovulatory signal originating from the pre-ovulatory follicle and or egg complex may synchronize sperm capacitation with oocytes availability and enhance their developmental competence. Okay. The above data support a central thesis. The oviduct does not passively store sperm, but actively selects and conditions a functionally superior subpopulation. To date, the relative contribution of sorting through binding versus modulation of sperm physiology via cell signet remains incompletely resolved. Sperm selection based on oviduct-inspired approaches remain largely confined to laboratory studies and have not yet resulted in commercially available selection tools for IVF. Although beyond the scope of this review, another promising application of sperm binding using oviduct ligands is a better predictor of male fertility in the future. Several studies have reported greater sperm binding capacities by oviductal cells in bulls with a higher pregnancy rate per insemination compared to low fertility bull semen, and that's not allowed a bull. Current sperm selection methods prior to AI or largely rely on motility or morphology-based approaches, which can only partially reflect the functional competence of the sperm. I'm actually needing to have a drink, I think. Expanding these strategies to incorporate the complex and finely tuned behaviors exhibited by spermatozoa within the female tract represents a promising avenue for improvement. In this context, microfluidic chips designed to sort sperm have recently yielded encouraging results for IVF, although their output remains limited and restricts routine application. So further, so our understanding of key female-derived interacting factors and mechanisms governing sperm selection remain incomplete. Bridging these knowledge gaps will be essential to develop more physiologically relevant and efficient sperm sorting strategies for assisted reproduction. So my comments, wow, what a lot to think about. This paper describes or suggests what happens during in vivo sperm preparation and interactivity between the oviduct, the sperm, and the oocyte that bear no resemblance to what happens in vitro. Some may say that it works just fine and current systems are okay. Even injecting a sperm into an oocyte, some think as normal. Yet so much happens in the zygote that set in train the process of allocating material to early cleavage stage embryos that direct specific cells into the ICM and beyond. Extracellular vesicles, in addition, appear to improve hatching as an example that a single embryo culture system under the lights and camera is normal. But to tell me this, why after 50 years our pregnancy rates remain so low? It's like trying to win a race when you are the only person in the pool. I think I need to stop there, have a cup of coffee or a beer. But I'd like to say that my name is James Stange. I've been an embryologist since for almost 50 years. I'm not a medical person, and this information is not medical advice, but it may help you in knowing what questions to ask about your treatment with your doctor, and maybe to help in understanding the advice to receive. If you like what I'm doing, press subscribe, press like, and leave a comment, and tell other people about this website. I do have to recognize that this is funded by my own ferday.com program, which is an online EQA and training program in IVF and andrology and embryology. And these are the different schemes that's involved in. And like just at this point in time, I really do need to stop. This is far too long. I'd like to thank you if you persevered to the end. I know IVF is a personal journey that involves the action of many techniques, ideas, and staff, and I hope this understands you on your journey. And let's not lie now to say thank you and goodbye.